Well, welcome everyone. This is Joanne Geer. I'm the Executive Director of the Westchester Biotech Project, and we really welcome you to the first session of our consortium on translational research in the microbiome, and you'll be learning a lot more of that very soon. And we've been working, I've been working with research on the microbiome for some time, but I will say that this whole consortium was really kicked off by Keith Bastian, who you're going to be hearing from in a moment. I'm just going to give you a sense of what we're going to be doing today. Please, if you are, when you've come in, please mute yourself. Is it Ajay or AJ? Would you please mute yourself if you could? If you're not talking, it's very helpful. Thank you so much. We are going to have a couple of presentations, and then we're going to go around, and we have quite a few people registered for today, so we may not get to invite everyone to introduce themselves, but we will try to invite as many as possible, but essentially to have some conversation about what's going on in terms of harmonizing enabling technologies in research, and to think about what we want to do next. The idea for this group is to be meeting on a fairly regular basis, online, and then ultimately in person. Chris, would you go to the next slide, please? So I want to introduce Keith Bastian, who's the CEO of the Institute for Life Science Entrepreneurship. Keith, I'm going to, I think you can unmute yourself and go ahead and speak.
Joanne, thanks very much. I'd like to thank you and the Westchester Biotech Project for arranging this web conference and for helping to lead in the formation of the Consortium on Translational Research in the Microbiome. As you mentioned earlier, the idea for this really rose out of Ilse's interest in microbiome research and our own entrepreneurial focus on translational science. As many of you know, ILSA is the Institute for Life Science Entrepreneurship. We're located in the New Jersey, New York metro area, and we're a regional life science accelerator with our own research labs, an incubator, a company that provides services in genomics, in microbiology, and we also run a number of regional events. Sam Kongsumet, who's on this call, is the head of the executive director of our Entrepreneur Center and has been very much involved in driving forward with this consortium. So as we all know, microbiome research is fast advancing with plenty of insights into the colon, skin, and other microbiological niches in human as well as other ecosystems. And since 2015, when ILSA became an active member in the field, and that was through our partnership and alliance with the American Type Culture Collection, dozens of microbiome companies have been formed with microbiome connections now broadly associated with both health and disease. Yet, we still do not have a true mechanistic understanding of the link between the microbiome in all these various niches in nutrition and the microbiome and disease. Furthermore, in basic microbiome research, there's a gap between what can really be studied using in vitro or ex vivo tools and what can be studied in vivo, which is where this complex ecosystem takes place. So it requires for the most part, in the case of humans, really studying the microbiome within humans, as the rodent models for translational animal models is really not, as we've learned, very predictive of what happens in humans. So this is further compounded by many technical challenges in validating and standardizing the enabling technologies for this research to work in genomics and bioinformatics, taxonomically, just to name a few. Microbiome research is subject to large variability between labs in how experiments are conducted and even the reproducibility of the data, which means that companies that are doing translational science and working in this space will need to create new technologies and systems for that validation, and probably to do this effectively, we'll need to work together to solve these problems. So the field has been, in large part, correlatory to advance the in translational science, it really needs to get to defining cause and effect and the underlying mechanisms. As I mentioned a minute ago, this requires standardization and standards and valid systems of study. So one area of research that we've been involved with at ILSA is the development of a translational animal model of the microbiome. And to do this, we're collaborating with animal scientists at the University of Wisconsin and Columbia University in the creation of a humanized gut microbiome translational model in the mini pig. And we've already found some really nice correlations between some disease states in genetically engineered mini pigs and what's seen in human clinical trials. So the basis for a valid translational model is there, and we're currently working to develop a system that would be as economically as possible with these large animals that we can make available to the research community as a test point, I guess, an evaluation point before going all the way from some in vitro model to a clinical trial in humans. So there are many needs that this consortium can address. We felt that a forum to address these translational issues is needed, not another meeting just to present on the company's findings and accomplishments, but to address the core issues in the enabling technology. So I guess that's the purpose here, the vision of how we're going to do this, what this practicum will look like, how we'll operate, and so forth, is an important introductory point that we should talk about. And I'll turn it over to Eric to sort of explain a little more how we envision working together. Thank you. If you have any questions, please feel free to ask.
Well, thank you, Keith. This is Eric Vieira. I'm the Director of Special Research Programs for the City University of New York. So where I sit at CUNY, as we call it, is in central administration. And it's my role here in central administration to look for unique ways to engage the research community to benefit our faculty and our students who may be wanting to explore very innovative new research spaces. And so when Keith and Joanne and I got together, along with members of the steering committee, who you will be introduced to shortly, thought about this, we definitely saw the need for both developing a virtual community in order to share knowledge and really build consensus around next steps in regards to research, particularly in this very new field of microbiome research. And the way we thought about constructing this was to really think about, how do we bridge both the physical and metaphorical divide between researchers, institutions. CUNY needs this type of approach, given the fact that we have over 270,000 students spread across 25 different colleges and professional schools and graduate schools throughout the 5 boroughs of New York. So getting anyone together at one time, providing a virtual means to do that will serve us very well. We also want to then explore, obviously, a hybrid where at times where we need to bring people together, we can do that in locations, in the East Coast, whether it be in New Jersey or across the river in Manhattan or one of the 5 boroughs, and we have the capability of holding those meetings given our presence in all the 5 boroughs of New York City. So along with the 270,000 students, we have a faculty of over 7,000. Many of them are in the life sciences that are exploring these types of fields. And therefore, this type of working group to really explore and focus on enabling technology to advance research is something that we believe in, and the approach is going to be significant to us. And that's why we're extremely happy to partner with ILSA and the Westchester Biotech Project in order to enable ourselves to be able to participate and also look at how do we advance microbiome research. So I'm really excited to be a part of this. and look forward to the engaging discussions that will take place today, as well as future virtual and in-person events that we will be organizing in the future. So I'll stop there. Thank you very much and look forward to the presentations today.
Thank you, Eric, and thank you, Keith. Chris, would you go on to the next slide, please? This is our steering committee. We've got a really wonderful and diverse community here, and yes, we are quite open to having new participants, so feel free to be in touch with us if that's something you would like to do. Would you go on to the next slide, please? In a moment, we're going to hear from Scott Jackson from NIST and then Dev Matar from ATCC. We really are excited that these are our speakers today because as we look at issues around developing standards, obviously, if NIST is leading the way, then we want to find a green space where we can support their work. And we really appreciate also Dr. Matar, who's on the ground running a lab where all this work is going to be relevant. We'll be hearing from some other people today as well. Would you go to the next slide, please? Just going to introduce the Westchester Biotech Project. We're based in Westchester County, New York, which is just north of New York City. And we really are focused on that developing collaborations across our region, but also nationally and internationally. So we're so happy to have folks here from around the world today. We're about to sign an MOU for a Westchester Biotech Project Europe group. So you'll be hearing some more about that as well. Would you go to the next slide, please? And these are some of the partners that we've been working with over the last few years to get to where we're going, and just wanted to give a thank you to them. Next slide, please. So you can, during the presentations, we ask you to put your questions in the chat box and we'll address them after the presentations. And we'll see how it goes in terms of inviting people to actually speak. We'll have about 10 people that we're going to call on, and I'm not going to warn you whether it's you or not. We've made some choices from the people that we see who are registered. We'll have a really great discussion after the presentations. Okay, you can go on to the next slide. There we go. So I'm going to introduce Dr. Jackson, and he'll present, and then he'll hand things over to Dr. Matar. And I'm going to make you presenter in 1 second, Scott. Just got to find you on the list. Got a nice group here. Almost there. There you are. There you go.
All right. Can you see my slides? And can you hear me?
Yes, we can. You sound great.
Okay. Okay, so I'll go ahead and start. Yes, thanks to the organizers, Joanne and Eric and Keith for getting this going, and we're certainly happy to be a part of it. We recognize the need for standards in microbiome space, and I'm going to talk briefly, just 10 minutes, about some efforts that are ongoing at NIST to address these issues around microbiome measurements. I'm going to throw a lot of information at you in a very short time, so like websites and dates and things like this. So if you want to take notes or take screenshots as I go along, I think it'll be helpful. Since I am not going to go in detail on any one thing.
Scott, we can certainly share all the slides if you're comfortable with that. We can put them right on our website.
Yeah, I'm happy to share.
And we'll send a notice out to everybody when they're available. Thanks.
Okay. So I feel obligated to introduce NIST because I know a lot of people aren't familiar with NIST. So quick background, we are a non-regulatory federal government agency in the Department of Commerce. Our headquarters are in Gaithersburg, Maryland, but we also have a large campus in Boulder, Colorado. We're the oldest technical institute in the federal government, dating back to 1901. We have about 5,000 scientists and engineers and 4 Nobel laureates that we're very proud of. And we have about $1 billion annual budget. And regarding Technical Institute and Department of Commerce, sometimes people see a disconnect in that. Why does the Department of Commerce need a technical institute? And it goes back to our mission, which is to promote U.S. innovation and industrial competitiveness by advancing measurement science, standards, and technology. So we see industry as our stakeholder. And in the microbiome space, I see my stakeholder community as all of the big and small companies that are beginning to work to develop microbiome therapeutics and diagnostics. So this is a highly regulated space, and in regulatory space, you have to, measurement science becomes very important, standards become very important. So I'm sure most of you know this industry is expected to grow over the coming decade. There are companies that currently offer a clinical diagnostic that measures the health of your gut microbiome, Ubiome, which I'm sure most of you have heard of. This is a real clinical diagnostic in the sense that your physician orders it, you send in a sample, and then you go to see your physician to get consulted with the results of the assay. And other companies are using metagenomics for infectious disease diagnostics in a very similar way. So what is NIST doing in this space? I'm going to talk briefly about 4 areas where we're active, those being interlab studies, reference material development, measuring ecological factors that govern microbiome function and stability, and stakeholder engagement, namely consortia and workshops. So one quick slide on the interlab studies that we've been involved in. A large interlab study that launched in May of this year, earlier this year. It's called the Mosaic Standards Challenge, and you can sign up to participate in this interlab study by going to mosaicbiome.com. And this was a Herculean effort that was put together in collaboration with four agencies and institutes, those being DNA Nexus, Janssen, BioCollective, and DNA Genetech. And what we've done here is put together a kit of reference materials that consist of whole stool and DNA mixtures. And what we are anticipating is that we send these out to labs around the world free of charge. We've made 700 kits, so it'll be, you know, 7 samples measured 700 times across the world. So as I mentioned, it was a Herculean effort to put this together, and it was mainly supported by Janssen. And NIST's contribution to this is ground truth DNA mixtures. So these are metagenomic mixtures of DNA that we've made, and we made 800 aliquots and sent to BioCollective for distribution in this challenge. So I encourage you all to participate in this. It is I think it's the reward that comes out of this is going to be immense in terms of understanding sources of bias and variability in the measurement process. And like I said, it's free of charge to participate. Any lab can participate, so please do so. The second bullet I wanted to touch on is a metagenomic reference material that we are currently developing at NIST. This project was funded by the FDA. It was this metagenomic reference material is specifically designed to assess the analytical sensitivity and specificity of next-gen sequencing-based pathogen detection. I say pathogen detection, we're talking about microbiome space, but fundamentally they are both metagenomic measurements, and we need to understand the sensitivity and specificity of metagenomic measurements. So the mixture or the reference material we are making consists of genomic DNA from 19 different microbes, and the idea shown schematically here that you can make quantitative and qualitative mixtures that allow you to assess the performance of your NGS-based metagenomic measurements. And we've been working on this for a couple of years. We've got all the material produced, and we're currently characterizing it. And in the time that we've been using to characterize it, we've also been making some mixtures just to show sort of a proof of concept what the material can be used for. So here we made log dilution mixtures of various strains and looked at expected outcomes versus what we measured. And like I said, I don't have time to go in any detail, but we're also able to make in silico mixtures that mimic our in vitro mixtures that give us even more control over the raw data that we're actually analyzing. And ultimately, as I said, what the FDA asked us to do is produce a material that allows them to assess analytical sensitivity and specificity of the metagenomic measurement. And by making mixtures like this, we can actually calculate and show sensitivity, precision, F1, which is harmonic mean, etc. And yeah, so that's all I'm going to say about that. The material is, we're sort of putting the final stamp on the certifications now. I would expect it would be ready for distribution early next year. And finally, a project that we've just become integrated in, I guess, is the EcoFab Consortia. EcoFab, as I mentioned, is a consortia. It wasn't started by NIST. It was actually started by Lawrence Berkeley Lab. But it is a movement to develop in vitro tools that allow us to manufacture reproducible microbial ecosystems. So in the simplest concept is that, you know, no two microbiomes in the world are the same. So how do we do reproducible studies of microbiomes when we don't have two that are the same? And the philosophy here is to be able to engineer, build microbial communities that can then be systematically studied using some in vitro device, like a microfluidic device that's shown here on the left. So I think this is going to be very important in the microbiome space, in that it will allow us to perform systematic studies on reproducible microbiomes. But you could also imagine that this might serve as ground truth, as reference material. If you can engineer reproducible ecosystems, then you have essentially a reference material, ground truth, for benchmarking your metagenomic measurements. And lastly, I just want to touch on stakeholder outreach. So it is very important for us to understand the needs of our industry, of our stakeholder community. So we work very hard to reach out to those people, those companies, industries that we see as our stakeholders. And we do that in two ways. One, we have a consortia. It's known as the International Microbiome and Metagenomic Standards Alliance. I encourage you all to become part of this. You can find the IMSA home at microbialstandards.org. Once you're on microbialstandards.org, you can sign up to be a member of IMSA. But I want to point out too that microbialstandards.org was designed to be what I call a one-stop shop for all things related to microbial standards, not just NIST efforts, but anything internationally that's related to microbial standards. We tried to represent it on this website. So you can see some of the headers here: reference materials, standards, organizations, bioinformatic resources. So as you're going to hear from the next speaker, Deb, is making some microbiome community reference materials, and we have those represented in our website here, as well as other reference materials for microbiome and metagenomics that are available. And lastly, I just want everyone to put a save the date on your calendar. In 2019, we're hosting a NIST workshop, standards for microbiome measurements. It'll be the week of June 24th, probably a 4-day workshop, located here at NIST headquarters in Gaithersburg. And while we're still very early in putting the agenda together, the thought now is that we will spend 2 days focusing on standards for metagenomic measurements of microbiomes. Day 3 will be standards for metabolomic measurements of microbiomes, and day 4 on data and metadata standards. So that's sort of the rough, broad agenda as of now. And we'll have more information on this workshop and a sign-up page coming online shortly. So that's all I have. I'll turn it back over to Joanne, or if there are any questions.
Thank you very much. I think we'll take all the questions at the end. And Eric, I'm going to make you the presenter. And I just want to say a word while I'm making this shift for our friend Chris Kinzel. So Chris is our program coordinator, and you all have received a good amount of communication from him. And I just want to say, without Chris's work on this from the very beginning, it wouldn't have happened. So I want to thank you for that, Chris.
Thanks. I'm humbled. Appreciate it.
You've done good. Eric, you should have a pop-up to make you the presenter, and you also need to unmute yourself. Oops, you know what? I may have sent it to the wrong thing. Hang on a second. We'll cut this out of the recording, right? No, you should be the right. I have you and the I have you logged in twice, once for your phone and once not. Let me try this again. Eric, would you just try unmuting yourself? Let's see if we can get that far. Ah, there you go.
Okay, I'm unmuted. Sorry. Yeah.
Do you see a pop-up to
Yes.
ask you to be the presenter? There you
Yeah.
go. Let's see. Oh, so there's some. I have to name check Howard Young, who's one of the original people who got me involved with the microbiome and who hosted a wonderful program for us a couple of years ago at NCI in Frederick. And it's really been a pleasure getting you involved with this, and very happy you're here. I know there are a number of people on the call who were invited by him. How you doing there, Eric? We had no trouble last time.
Aren't we going to Dev right now?
Oh my gosh.
Sorry.
Well, you
Dev will be
were all thinking, what is she doing? It's okay to say that. Dev, would you unmute yourself? You're going to receive.
Okay, I'm ready.
There you go, and you're going to receive a pop-up. Sorry about that.
All right. Can you hear me and can you see my screen?
You sound great. Thank you.
Excellent. Well, first of all, thank you very much for providing this opportunity to present some of our work that we are doing at ATCC. I think everybody knows about ATCC, but seeing Scott, I thought I'll add a slide for ATCC as well. So we are more than 90 years old organization, and we help researchers by providing controls and standards. We do have collection of more than 100,000 bacteria, fungi, cell lines, and we are based in Manassas, and we are a nonprofit organization. So the way, since I have only 10 minutes and I have 28 slides, so I'm going to drive a little bit over the speed limit here, but you're welcome to ask me any questions offline as well as after this presentation. So essentially, I'm going to show you some of the work that we have done by developing the MARC community standards, and essentially how do we QC and how researchers are using those standards. One thing I want to mention is we are not involved in standardization or development of assays. We do use existing assays to do proof of concept data, but we are not, I'm not necessarily going to show you the best data, and I'm not going to recommend any specific kit or protocol or any instrument. So I think Scott has laid down it perfectly, so I'm going to skip this. This microbiome research, everybody knows that it is heading towards clinical trials. The two methods which are popularly used for microbiome analysis are 16S ribosomal RNA profiling and shotgun metagenomic sequencing. Each method has its own disadvantages and advantages. Regardless, the methods are long, cumbersome, multi-step, and each step can introduce bias. And to really standardize the microbiome methodology, there is a solid need for a controlled, predefined, standardized reference material for assay development, verification, validation, and as a QC. So that's the need we are fulfilling at ATCC. We have developed MARC communities, which are a mixture of whole cells and mixture of genomic DNA, which mimic the microbiome specimen. And we also have done some work and data analysis as well. As you can see, this workflow is complex, and you can use the standards to optimize any step in this workflow. The way we build MARC communities is we pick up pertinent strains and we mix those strains in appropriate amounts as whole cells as well as genomic DNA. The strain selection criteria, although sounds very trivial, but there is a whole lot that goes into consideration while picking up the best strains and making a very diverse MARC community. Then we have two types of standards. One is general standards, which where we have mixed bacteria, very diverse set to challenge assay optimization and assay development folks. And recently we have started building site-specific standards which are geared towards oral, skin, gut, and vaginal microbiomes. And all of our products are bundled with a analysis platform offered by OneCodex, where you can take the data and you can get QC data directly in OneCodex. These are our general standards. We picked up on BEI MARC communities, which were offered as Human Microbiome Project, and we improved it, and we made 6 standards out of them. The bottom one are a couple of collaborations that we did, one with our distributor in LGC for making a pathogen mark community. And then we also worked with Scott Teague, who is chair of ABRF Metagenomic Research Group. And we made some standards based on their extreme microbiome project. The next one are the site-specific. This is what we are currently working on. We just launched oral and skin microbiome standards. These contain the bacteria which are prevalent in these normal and disease state of these sites. So these are more geared towards QC and clinical trials where you can run them, where the bacteria are more pertinent in that specific site, and you can run them as a daily run quality control standard. The next section is essentially how we QC our standards, which is quite challenging actually. So we use both 16S and shotgun metagenomic analysis for QCing our standards. This is an example of two products that we have. We do see a lot of variation with existing methodology. As I mentioned, the goal is to identify all the bacteria in the mix and to show that, okay, they are present in even composition. So the left side is what we were expecting, and the right are 16S and shotgun data from two mark communities that we mixed. And this is genomic DNA. Genomic DNA is easier for us because it's more controlled. We all get a lot of challenges when it comes to the whole cell because of the DNA extraction. Although DNA extraction sounds very trivial, but it is one of the most challenging steps. The data that you're seeing here is from a mark community with 3 different kits, and you can see a whole lot of variation in the final output of your relative abundance with 3 kits. Saying we run this mark community data first, and after that, seeing this variation, we took individual bacteria and we ran through the same kits, and you can very well see there is a huge variation among 3 DNA extraction kits for individual bacteria in the mix as well. So which really shows the value of these standards. How can you use to standardize each step such as DNA extraction? Same thing, we recently made an oral microbiome standards where we put together 6 bacteria. The genomic DNA on the top looks pretty promising. We can get the same ratios that we were expecting. But when we go for the whole cell mix where we tested 2 extraction kits, we get really a lot of challenges in terms of the relative abundance, whereas with genomic DNA, it's more controlled and easier for us. So definitely, these standards are really useful for comparing various methodologies here. We also use a digital PCR to determine precise genome copy number. For example, this is one of the standards which we have mixed pathogens. This is mainly for clinical metagenomics or for pathogen detection. Here we use digital PCR, which works pretty well for precise definition of genome copy numbers. This slide shows our batch-to-batch variability. So whenever we make a different batch of distribution lot, this is one of the products which has 4 different concentration of genomic DNA, and the gray dash is what we were expecting, and the yellow and the blue are different batches, and we see both batches fairly consistent as far as 16S and shotgun data. Our standards are platform agnostics. Most of our data is with Illumina, but we do have data from PacBio, IronTorrent, and MinION as well. The blue line is what we are expecting, and the other colors are MinION on the left, PacBio in the middle, and the IronTorrent is on the right. So we got fairly consistent data with all platforms. So basically, our mark communities can work with any sequencing platform. How customers are using our products, some examples. For example, this data was generated by Stefan Green from UIC. This is one of the products that we developed with ABRF. So these are 3 different runs done by different time, and we see fairly good consistent data from different runs. This is using different Illumina platforms. Again, Stefan Green from UIC ran this on ABRF standards. MiniSeq, MySeq, NextSeq, HiSeq, all those platforms run differently, but they produce the same data sets actually. Then this is the data which we recently got from PacBio. They ran our mark communities and using the full-length 16S ribosomal RNA profiling and shotgun as well. we see a very nice correlation of expected versus observed, and this is the analysis that we offer. We can very well see all true positive 100% score in our analysis as well. This is the data from Dr. Pat Gilbert, who is our neighbor here in George Mason University in Manassas. And he ran our standards on IronTorrent using both 6-NIST and shotgun. And the data was not perfect as we expected, but it is pretty consistent in terms of identifying all bacteria in the mix. These are all standards that we have, and I'm going to give you a quick glimpse of what we are currently offering. We are now working in R&D on a spike-in standard where we have modified a 16S ribosomal tag, and this unique tag would be integrated into bacterial chromosome. We've already produced a couple of bacteria with those unique tags, and we spiked in. For example, here is the E. coli in different quantity of spike-in, and we can see this recovery as dose dependent of spike-in. We'll be offering this product hopefully early next year. And we are also working on a fungal moth community microbiome standards. We have run a couple of beta trials on this. We could get very consistent correlation of what we put versus what we observed with shotgun metagenomic sequencing. It has 10 fungi. And then we are also making a virome standards where we are trying to mix viruses, which is more challenging because we have a lot of background RNA, DNA from viruses when we grow viruses in the host cells. But this is still early. We have made libraries using both RNA and DNA, and blue is mostly the background DNA or RNA coming from the cell lines, but we could still able to identify all the viruses that we put in the mix. To summarize, this is what our portfolio looks like. We have moth communities, which are general standards, and now we have started offering site-specific standards for skin, gut, and vaginal and oral microbiomes. We are pretty close in delivering to the spike-in standards as well, and then we are working on virome and microbiome as well.